Good evening and for all the people in Indonesia and in South Africa. And good afternoon to people who are seeing this from France or from Europe. It is a real pleasure to welcome you for our second scientific webinar that will be about the coronavirus. So before getting started, we will hear a few words by our scientific attaché in IFI, Mr. Thierry Marais. So Thierry Marais, you can hear me.
Thank you. Thank you. Selamat sore. Bonjour. Good afternoon or good evening, everyone. First of all, I would like to say that we are very pleased to welcome at this webinars. In the Scientific Cooperation Department in the French Embassy in Indonesia, we aim to promote bilateral scientific and technological relations between France and Indonesia by offering scholarships for Indonesian students who want to pursue their PhD in France, by financing collaborations between France and Indonesian researchers. We also give importance to popular science and regularly invite researchers to talk about their work. And whenever possible, we try to focus on the news. The ongoing news at the moment that we have been dealing with for the past few months, as I am sure that you may all familiar with, is the coronavirus. But do we already know what is coronavirus? What is the difference with the common virus? Where does it come from? How did it emerge? How does it spread? Why do we need to do the lockdown as well as social distancing? Will it be vanished one day? To answer all of those questions and many more, we are pleased to welcome a world-renowned virologist. But before that, I will let my colleague Miriam to introduce the speaker to all of you. Again, thank you for being here, and I hope you will enjoy the presentation.
Okay, so thank you so much for your intervention, Thierry. So like Mr. Marais said, we invited a really renowned virologist, Mr. Thomas Mourez. He is the current health attaché in the Embassy of France in Vietnam. He used to be a lecturer in the Faculty of Medicine and the Hospital of Rouen in France, and his research aimed to study the adaptations of animal virus in humans. So the least that we can say about Thomas Mourez is that he knows the coronaviruses. Indeed, in 2001, he has done his thesis on coronavirus respiratory viral infections. And since then, his work in the field of coronaviruses has won him almost 500 citations in international scientific papers. So Mr. Thomas Mourez is a doctor of pharmacy, a doctor of science, and has a university diploma in medical pedagogy. Before starting the actual conference, I would like to remind the audience a few technical details. So after Thomas Mourez's presentations, we will have a short question and answer session. So you are invited to ask your questions in the Q&R tab on your Zoom app. And you can also interact with each other or simply react to the presentation in the chat tab. For people who are currently following this webinar on our Facebook page, please feel free to react, comment, and ask questions in the comment section on our Facebook page. We will also take questions from there. So I think I said everything. And Mr. Thomas Mourez, the floor is now yours.
Thank you, Miriam, for your kind introduction. Good evening to everyone. I wish that you can hear me well. I'm very pleased to speak about the emergence of coronaviruses this evening, since, as you said, as a virologist, they were the first topic of my research 20 years ago. When the scientists speak about the emergence of viruses, it usually covers three main different concepts. The first one is the discovery of the viral origin of an already known disease. One example can be the discovery of the role of papillomavirus in the development of the cervical cancer by Harald Zuhausen, who got the Nobel Prize for his discovery in 2008. The second concept, when we speak about emergence, is an epidemic of a known viral disease with abnormal epidemic character or a change in the geographical distribution. One of the best examples could be the introduction in New York in 1999 of the West Nile virus, a known African virus that spread among the United States in a very few years. And the third concept of emergence covers the creation and the dissemination of a new virus in a given population. The best example here is the emergence of HIV in Cameroon and its dissemination all over the globe. The SARS-CoV and the influenza H1N1 in 2009 are also good examples of this concept. So in 2019, the emergence of a newly highly pathogenic virus, the SARS-CoV-2, responsible for the current COVID-19 pandemic, has been described. What we observe is that these events are more and more frequent, and not only because they are publicized by mass media. At this occasion, we discover, or we rediscover, the family of coronaviruses with a strong potential of emergence in the human and the animal populations. So, after a few simple virology reminders, I suggest that we discuss the elements necessary for the emergence of viruses in a population. We draw up an overview of the known coronaviruses in animals and humans to explain the successful emergence of this new epidemic. And we recall the major societal and environmental changes that have seen the multiplication of emergence events. So let's start with few informations on viruses for those of you who are not used to them. The viral particles are composed of a limited number of different structures. The first one is a nucleic acid, the genome, of only one type. It's either a DNA or an RNA. There are usually enzymes which participate to the replication and maturation of the virus. There is also what we call the capsid, which is a protein structure with different shape, icosahedral, like this example, or sometimes helical. And in some viruses, like the coronavirus, we can observe an envelope derived from cell membranes that surrounds all the other elements. The spikes present at the surface of the envelope that we can see here allow the virus to bind the target cells of the host, and it is also the main target of the immune system when the host develops neutralizing antibodies. The purpose of all these structures is to protect the genetic information of the virus, to target the cell to infect, and to replicate the virus. The DNA or the RNA nature of the viral genome is crucial. The DNA viruses, like the herpes virus, have the possibility to present a very large size and therefore hundreds of genes, but they have a low potential for genetic evolution. On the contrary, the RNA viruses, like the coronavirus or influenza, they have a relatively small size and limited number of genes, but they have a very high capacity for variation by diversified mechanisms. All things together, we distinguish in virology naked virus with an icosahedral or helical capsid, enveloped virus with the DNA or an RNA genome, and some exceptions, because we are in medicine. The coronaviruses we will focus on this evening are enveloped virus. They present They present an helical capsid, you can see over there, with a large RNA, one of the largest known in the RNA viruses. The human coronavirus, that we call the HCOV, and the animal coronavirus have all the same structure. As I said before, the virus have the capacity to evolve, and the genetic evolution of viruses can be explained by minor mutations in a few bases, but also by major changes in whole sections of the genome. Certain viral families, like the coronavirus, have great capacities to evolve by various mechanisms, by point mutation. I'm sorry, I'm not used to the... by mutation, by insertion and deletions, and by exchange of genetic materials, and especially in the coronavirus through recombination. So there is also an important principle in virology, which is the species barrier. A virus generally is only adapted to one single animal. A virus that is infecting a cow only infect cows, and the virus is circulating among the population of cows. Usually, it's not possible for a cow virus to infect humans, monkeys, mice, or other species. So this is an important element in virology. But it happens that in certain rare circumstances, there can be accidental transmission of an animal virus to humans, where humans are usually deadlocks. It is called a zoonosis, as we can see usually with rabies. In the case of rabies, the virus circulates in dogs, especially, but also in wild carnivorous mammals, and the virus sometimes can be transmitted through biting to humans. In case of infection, the patient will die from rabies, but there is no subsequent transmission to other human beings. The patients are deadlocks. In even rarer cases, the virus is able to cross the species barrier and adapt to its new host by specific evolutionary mechanisms. That is the phenomenon of emergence. The best example we have is the emergence of HIV. In Africa, for thousands of years now, the SIVs, which is the simian immunodeficiency virus, the SIV have been circulating among a wide range of different monkeys. And probably at the end of the 19th century, the virus crossed the species barrier from an infected chimpanzee to a human. The virus adapted to the human, and the dissemination among other humans started, resulting in the HIV pandemic we know since the 1980s. So let's now say a few words about the coronaviruses that infect the humans. 7 coronaviruses are described in the human population. We describe 4 classical coronavirus, which are ubiquitous and responsible for upper and lower respiratory infections, generally mild in the immunocompetent individual. They are known as the coronavirus 229E, NL63, OC43, and HKU1. Fatal cases are sometimes described in immunocompromised patients, but it's a rare phenomenon. There are also 3 emerging coronaviruses that are described. They are highly pathogenic viruses which emerged in the recent years. The SARS coronavirus that emerged in 2002, the MERS-CoV, which emerged in 2012, and the SARS-CoV-2, who emerged in 2019. For these 3 viruses, severe infections are described, especially in the frail persons, but also in the subjects considered to be in good health. So, after these few elements about the viruses, let's speak about the main actors and elements that are required for the viral emergence. The emergence and spread of viruses depends on three main factors: the virus, the host, and the environment. The host must be susceptible to the infection, which requires no pre-existing immunity, an adapted route of infection, for example, respiratory infection, enteric or sexual infection, sexual route of infection, and the presence of at least one binding receptor at the surface of the host cells. The host, to be able to spread the infection, must also be able to spread, to excrete the virus. The virus must be adapted to the host, and usually the new viruses evolve from an existing virus in the same host. So, for example, a new human virus can evolve from an existing human virus, but sometimes it can also be adapted from an animal host. A strong capacity for evolution is also required to increase the probability of an emergence phenomenon. Finally, the environment must favor the contact of the host with the virus. It must favor the transmission among subsequent infected hosts, and it must favor the local, national, or international spreading of the virus. And in that, the social cultural habits are essential at every stage. So after explaining these few elements required for the emergence of viruses, let's explain why the coronaviruses have a great potential of emergence. Why coronavirus have a great potential of successful emergence? Because of the wide diversity in many reservoir animal species. To date, there is an increasing number of diverse coronavirus described mostly in mammals, but also in birds, as we can see on this phylogenetic tree. Here are the different viruses, and here are the hosts. So here you can see that they are essentially mammal hosts, and sometimes it can be birds. So the first description of a coronavirus occurred in 1937 with the discovery of infectious bronchitis virus in poultry, which name is now the avian coronavirus. And the first human coronavirus were described in the 1960s. Why coronavirus have a great potential of successful emergence? Because of their organization in quasi-species. When an individual gets infected by a virus, the diversity in a single individual at the beginning is really poor. But as the virus replicates, few mutations appear in many viruses, creating what we call a quasi-species. So each individual has a set of variants whose composition is in dynamic equilibrium, constrained by the evolutionary forces of the environment, and the presence in an individual of a multitude of viruses, all slightly different, increases the chance of having a virus capable of adapting to a different host. Why coronavirus have a great potential of successful emergence? Because they can cross the species barrier. As we have seen, there is a large animal reservoir, especially in mammals, that are potential candidates to cross the barrier, and in some viruses, they can exploit common cell receptors present in different animal species. If we take the example of all the known coronavirus infections in humans, one suspect an animal origin, all of them resulting from one or multiple species barrier crossing. For 5 out of 7 human coronavirus, the most likely animal origin is represented by the bats. The viruses were introduced into the human population via an intermediate animal reservoir, we can see here, most often the farmed or companion species introduced in the environment of humans. It can be cows, it can be dromedaries, it can be civets or other animals. The introduction of coronavirus in the human population is a regular phenomenon. As you can see for NL63, the introduction is probably dating from the 13th century, though the SARS coronavirus 2 has been circulating since probably the mid-2019. So let's take an example, speak about an example with SARS coronavirus. The SARS coronavirus circulated from November 2002 to July 2003, starting from China and spreading in 32 countries. Nearly 8,500 patients were infected and 850 patients died from the infection. This is the first epidemic in the human history for which the story of the emergence could be fully reconstructed historically, geographically, and genetically. When the scientists worked on the origin of SARS, the first investigations showed an epidemiological link of the infected patients with a live animal market in Guangdong as an interface. The serological surveys demonstrated a higher seroprevalence in the people working with animals compared to the other people. So at that time, samples from different animals on the live markets were tested, and the virus was described at least in two different species, the raccoon dog, you can see here, and the civet. The fact that several species sold on the markets were infected by SARS coronavirus suggested that they were only intermediate hosts contaminated during the passage through living markets, causing the human contamination. So, in the following month, the scientists from all over the world sampled a large number of animals to discover the reservoirs of the virus. You can see here one of my colleagues in my lab, Meriadec, on a field trip to sample the wild animals. And finally, the samples taken from a population of rhinolophus bats have shown that they were the main reservoir of the virus. All the work done has demonstrated that SARS coronavirus infection in humans had a unique origin linked to a very limited number of species barrier crossing from the bat to the civet and then to humans at the end of 2002 in China. So let's come again to our question. Why the coronavirus have a great potential of successful emergence? Because they can use analog cell receptors on different species. The study of SARS-CoV-2 is therefore really interesting for that. The virus binds to a specific receptor called ACE2, which is largely present at the surface of human cells, especially on the blood vessels. Other coronavirus, human coronavirus, also use the same receptor, like the SARS coronavirus and the coronavirus NL63. The study of the spike of the SARS-CoV-2 has demonstrated that the binding site of the virus was able to interact with ACE2 receptor of a wide range of mammals. On this publication from Zhao, published in Nature recently, the authors have transfected HeLa cells with ACE2 receptor from different animal species to check if the SARS-CoV-2 was able to infect their cells. The cell nucleus, you can see here appearing in blue, and the presence of ACE2 receptor at the surface colored in green. Here you can see the presence of the virus replicating in the cell that appears in red. So this study has shown that the SARS-CoV-2 was able to infect cells from both humans, here, bats, pigs, civets, but not the mouse. Other studies have shown that a broad range of mammal cells can also be infected with this virus. So let's come again to the question, why coronavirus have a great potential? Oh, I'm sorry. The ability of the SARS-CoV-2 to infect the cells of many mammals pose a potential risk of infection to new animal species. And the outbreaks we have seen around the world in slaughterhouse requires continuous attention from the health authority in case the SARS-CoV-2 that is circulating in humans can cross again the species barrier. So let's continue with the question, why coronavirus have a great potential of successful emergence. It can be because of the plasticity of the genome, which tolerate the large insertions or deletions. During the replication, the RNA replicating enzyme can slip and shift, causing an insertion or deletion of one or more base in the genome. As you can see on this drawing, the virus can survive in case of a deletion of a part of the genome or in case of an insertion, you can see here, of an additional fragment. The consequence of these insertions or deletions is the synthesis of a chimeric protein different from the normal protein with altered properties. To illustrate this fact, let's take an example in pigs. The transmissible gastroenteritis virus, which is called TGEV, is a coronavirus which has infected the pigs since the 1940s, causing very significant economic loss in farms in Europe and the United States. The mortality in piglets under 2 weeks old are usually about 100% of mortality. This coronavirus has a marked enteric tropism causing the characteristic diarrhea. In 1984, routine serological checks carried out in Europe revealed a sudden and very significant increase in the seroprevalence of TGEV in pigs, while no increase in diarrheal episodes was suggested of a virus epidemics. This observation led to several samplings campaigns and the isolation of an emerging virus, which was called the respiratory porcine coronavirus, PRCOV. Several hypotheses explaining this emergence has been studied at that time. The immunization trial with attenuated TGEV strains in Eastern countries in the context of Cold War, where the scientists from the East were not sharing their results with the scientists coming from the West. The recombination with another animal coronavirus, accidental crossing of the species barrier from an unlisted wildlife coronavirus. But finally, the comparison of the genome of TGEV and PRCOV showed the existence of a large deletion, you can see here, of nearly 700 bases of the spike gene, leading to the formation of a truncated spike protein. This deletion led to a profound change in the tropism of the virus, leading to a modification of the pathogenicity. The respiratory virus was less pathogenic than the virus causing diarrhea, and of the route of transmission from enteric to respiratory transmission. In a few years, the emergence of PRCOV has acted as a natural vaccine against TGEV and has greatly reduced the TGEV infection by a phenomenon of cross-immunization. The cross-immunization is the fact that antibodies can protect against the two different viruses. Well, let's come again to our main question. Why coronavirus have a great potential of successful emergence? Because they can recombinate with each other. In some cases, the genetic evolution of viruses can be done by exchanging large fragments of that genome. The genetic recombination is the formation of a chimeric genome composed of several genomic fragments of different parental origin. It leads to the production of offspring with a combination of different traits that differ from those found in either parental virus. To make it clear, let's have a look to this illustration. In case of double infection of the same cell, the RNA replication of each virus is carried out independently. During the replication of the red virus here, the polymerase in green can stop in progress and change matrix to slip from the red RNA matrix to the blue RNA matrix. You can see it here. The polymerase then continues to replicate the RNA, generating a hybrid RNA whose beginning of information here corresponds to the first parent in red, and the end corresponds to the second parent in blue. The resulting chimeric genome is packaged and then released to infect new cells. So you have at the end viruses similar to the first parent, similar to the second parent, and different from both of them with a chimeric genome. Let's look at the case of SARS-CoV-2. The virus probably comes from bats. The SARS-CoV-2, which infects humans, results from the recombination of two bat cov, you can see here, and one pangolin cov, which would have taken place either in bats or maybe in pangolins or in another intermediate host that we don't know. After the crossing of the species barrier, and probably under the effect of natural selection, it then spread to human. In the condition, if the condition of the emergence of SARS-CoV-2 have not been highlighted to date, all the evidence converge towards the natural origin of this new infectious agent. The emergence of SARS-CoV-2, like in most zoonotic infections, is probably linked to anthropization. Anthropization is the transformation of ecosystems under the action of man, for example, the deforestation, which promotes the encounter between several animal species and humans, when this phenomenon is normally completely unusual. The role of markets for wild animals is also a serious hypothesis. So, after the description of the properties of the coronavirus that could explain the recurrent emerging success of this family of virus, I would like to speak about the role of the social habits in the risk of emergence. And for that, let's take an example with the Merscorinovirus. Unlike the SARS-CoV we have described at the beginning, the Merscorinovirus currently circulating in dromedaries and humans in the Arabian Peninsula have multiple origins corresponding to independent and repeat crossing of a species barrier and poorly effective interhuman transmission. On this phylogenetic tree, we can see that this virus has circulated since its emergence in humans in 2012, both in dromedaries, you can see in green, and in humans, you can see in blue, in a zoonotic manner. Indeed, no founder effect is observed on this phylogenetic tree, supporting the idea of a single transmission of a dromedary virus in humans. These repeated events can be explained by specific sociocultural habits in the region. In the Arabian Peninsula, the dromedaries are more numerous than humans. The people have, by tradition, very close contact with these animals, as we have in the Western countries with cats and dogs. So the most credible hypothesis of this recurrent transmission is the close contact with the animals or the consumption of milk directly at the udder of the dromedaries. As the saliva and milk contains numerous infectious viruses in infected animals, the contamination would occur through this route repeatedly. So close contact with dromedaries or consumption of milk from the dromedaries. Thus, the proposed epidemiological chain is as follows. The suspected origin of Merskoronavirus is in the population of bats. It happened that the virus was transmitted to dromedaries and emerged in the cattle of the Arabian Peninsula. Then, repeated zoonotic transmission of Merskoronavirus from dromedaries to humans are ongoing since 2012, with a limited human-to-human transmission in the general population. But for a reason we do not understand yet, the virus is efficiently transmitted among humans in hospitals and healthcare settings. The Merscorinovirus epidemic is ongoing since no public health intervention are done to date on the animal reservoir. Hopefully, when the virus was introduced in foreign countries, like South Korea in 2015, the containment measures around the cases were really effective. So the Merscorinovirus is not a pandemic yet, but it remains that there is still a high risk of pandemic in case of mutation of the virus and a better adaptation to human. I would like to finish my talk by questioning the role of the global changes in the multiplication of successful outbreaks. On a human scale, major historical transitions are defined, all of which have resulted in societal and environmental changes, as well as in exposure to various pathogens, favoring their emergence in the human population. 5,000 to 10,000 years ago, the agriculture and livestock development has, for example, greatly increased the exposure to domestic animal reservoirs. During this transition, the chance of seeing enzootic pathogens having contact with Homo sapiens increased enormously. It was probably at this time that the precursors of influenza, measles, chickenpox, and other smallpox emerged, and especially after the domestication of cows or chicken. The European imperialism and the great exploration that started in the 15th century are another key historical transition that favor the dissemination of virus. These transoceanic expeditions allowed the dissemination, the spread of most of the human disease in the naive population, and the dissemination of disease in America through the conquistadors or the slave trade, or in the Asia-Pacific region, has probably played a major role to help the European armies to conquer these territories. Today, the humanity is experiencing a new major transition in its history. The past few years have seen the globalization of world trade with the development of high-speed intercontinental transport, the galloping urbanization and deforestation, which destabilize the ecosystems, which favors unusual encounters between hosts, reservoirs, and microbes, and an uncontrolled demography, which increases the potential frequency of contacts. The current period is the one which concentrates the most societal and environmental factors that are favorable to the emergence and spread of the viral infections in the human populations, but also in animals and plants, and for the same reason, sadly. My last example to illustrate my presentation will be the SARS coronavirus. The epidemic, which started in November 2002, unfolded in 3 phases. The early phase that lasts for 3 months has seen a series of few independent cases, essentially in the Pearl River Delta in China, in the south of China. The intermediate phase that lasts for 2 months have seen the first superpropagation event in hospitals, and especially nosocomial epidemic in Guangzhou with 130 cluster cases. And the late phase that lasts for 6 months has seen the global spread of the epidemic after the superpropagation episode of the Metropole Hotel in Hong Kong. On February the 11th, 2003, the epidemic of SARS went global due to a single documented super spread event at the Metropole Hotel in Hong Kong, where a patient, which was in the room 911, infected 21 secondary cases in the hotel, of which 5 international travelers that subsequently spread the virus in Hong Kong, Singapore, Vietnam, and Toronto in Canada. In a few days, with the help of high-speed intercontinental air transport, the virus spread from China to 32 countries on the 5 continents. Hopefully, an early response and lockdown measures have permitted to end the epidemic few months after its beginning. Only one resurgence was described in 2004, when the virus accidentally infected a scientist in a Chinese laboratory who subsequently infected members of her family. This is now nearly the end of my presentation. I would like to discuss a few things before my conclusion. We have seen that the coronavirus are a large family of viruses that circulates in humans and animals with recurrent successful emergence. It is largely due to their biological properties that promote their evolution and adaptations in humans. They have a large RNA genome supporting the occurrence of deletions or insertions. They have a high potential for point mutations. They have a high potential for recombinations, and they have diversified tropism and diversified routes of infection. The emergence of epidemics required environmental conditions and socio-cultural habits that favors the encounter between several animal species, humans, and their pathogenic agents, when this phenomenon is usually completely unusual. There are many explanations, among them the anthropization of ecosystems, which bring together wild animal populations and humans, and especially through deforestation, or the sale of multiple species of wild animals on markets, especially in China and Southeast Asia. And we cannot dismiss the role of the use of wild animals in the traditional medicines. Finally, 2 elements are important to explain the growing number of successful epidemics. The major role of overpopulation, with the need to breed more and more animals for the protein consumption, production of meat, of milk, or eggs, and to increase the size of cities and population density, which promotes the transmission of both the zoonotic and human-to-human infections. And the last element is the major role of the international travels. The 3 human coronavirus outbreaks these past 20 years have underlined the major role of international travel in the spread of viral infections, and the increase in the trade relations between all the countries of the world has increased the speed with which the virus can spread and has greatly complicated the global response to contain the epidemics. I would like to finish my talk with some things that we have shared with my colleagues last week. For years, among the specialists, the question has not been whether, but when a new emergence of coronavirus infection will occur, considering their high biological potential and favorable environmental conditions. Among all the continents, Asia have all the characteristics of biodiversity, agronomic intensification, and livestock farming, and integration into the international trading and touristic market, favorable to the emergence of new infectious disease with pandemic potential. So there is a need for major interventions from both the governments and populations to prevent the next one. Thank you very much for your attention. I would like to finish the presentation with acknowledgements to the Embassy of France in Indonesia for their kind invitation, and especially Professor Thierry Marais and Myriam Ameg. And I would like to thank also my colleagues from Normandy University in France, Mériadec Arguy and Professor Astrid Vabre, who is my mentor. Thank you again. I'm ready to answer your questions.
Thank you so much for your presentation, Thomas Mourez. It was really interesting. And we have some interesting questions too. So I'm going to read them out loud so everyone can hear the questions. But Thomas, you can also see them on your computer.
Okay.
So we have a first question by Rosita Wijojo. She asks, they are influenza vaccines already. How effective is the flu vaccine against COVID-19, regarding that COVID-19 is also a respiratory virus like the influenza? Does it protect us to some extent or not at all?
So the question is quite easy to answer. In fact, when you produce a vaccine, the goal is to produce antibodies directed to the spike of the virus. So the spike is the protein that it is at the surface of the envelope of the virus. So when you inject the flu vaccine, the purpose is to produce antibodies that will fix the spike of the flu, or the flu. The spike of the flu virus, the influenza virus, is completely different from the spike of the coronavirus. So the antibodies that are directed on the spike of the influenza cannot bind to the spike of the coronavirus. So there is no efficacy at all of the flu vaccine against the coronavirus. There is a need for a proper vaccine.
Okay, thank you for your answer. Next question, anonymous question. So the person asks, with the current new normal protocols implanted in Indonesia, which do you think will develop faster, humans acquiring immunity or viral mutation?
So, it's really difficult to answer a question. It's not no or yes. In fact, it's possible that the situation will not be the same in all the islands in Indonesia. It's possible, it's still possible to stop the epidemic in some places, on some islands on the country. We have seen that the virus has disseminated all over the world, so it's not possible to stop the epidemic. But what we have seen is that in some countries, in some places, it's possible to stop the epidemic. So probably in Indonesia, a part of the population will get infected and will acquire immunity. But it's possible that in some other places in Indonesia, the virus can be well contained, And in fact, the population will not be infected, and there is probably hope that this population can be vaccinated before they are infected.
So next question by Al-Fajri, a vet student. So two questions from that person. First one, is there any significant differences between the naked and not naked virus in terms of transmission risk and the infection that it made to the hosts? And second question, how can we know the difference between the sickness caused by the coronavirus and other kind of sickness, for example, pneumonia and chronic bronchitis in terms of early diagnosis?
So the first question is quite easy to answer. Usually the naked virus can stay in the environment for quite a long time. In fact, in the naked virus, the part of the virus exposed to the environment is made of protein. It's a protein structure that is really tough. So this naked virus can remain in the environment for quite a long time. They can, you can find them in sewage, you can find them in earth, you can find them at the surface of vegetables. So there is a lot of viruses that induce diarrhea, for example, that are naked virus. And usually the enveloped virus, because of their lipidic nature, they are quite fragile. So they are really sensible to detergent, to UV. So usually the enveloped virus requires very close contact from an infected person and a naive person to be infected. So it is the case with the coronavirus. You need to have close contact between the people to allow the transmission of an infected person to a naive person. Coronavirus are not as fragile as other viruses, but compared to, for example, enteroviruses, they are really fragile, and there is a need for close contact. So the transmission of this kind of virus is really effective usually through the respiratory routes, or for example, the sexual routes. Herpes viruses, for example, are well transmitted through sexual routes, and they are enveloped virus. The second question, how can we know the difference between the sickness caused by the coronavirus and other kind of sickness? I'm not a radiologist, but I know that the radiologist can clearly, easily distinguish the images through CT scan or RMI. I think it's CT scan, because the image of the pneumonia caused by SARS-CoV-2 is really typical from this infection and really different from all the others. So I think it's maybe the easiest way to distinguish the two infections. There is also something that is really particular in the coronavirus infection by SARS-CoV-2 is the loss of the smelling. It is not described with all other viruses. So this is maybe one element that is in favor of the COVID-19 compared to other pneumonia or chronic bronchitis.
We have a really interesting question. I think we all wonder that. Why almost all coronaviruses are from bats?
In fact, they are not all from bats. As I said at the beginning of my, or middle of my presentation, the human coronavirus 5 out of 7 of them probably come from bats, but some of them come from mouse, rodents, maybe rats, we don't know exactly. And I would say that in animals, they don't come from bats all the time. So it's a specificity here that the human infections come from bats, but it's not mandatory. So they don't come all from bats. They can come from other species.
Okay. Another question. Is there a risk of recombination between the SARS-CoV-2 and other human coronaviruses? If so, what could it be the impact on the transmission?
So it's always possible to have a recombination between 2 coronavirus. It's one of the main properties of the coronavirus. But the consequences are difficult to preview, to guess. As we have seen in the example of PRCOV and TGEV in pigs in Europe and in America, you can have an increase in the pathogenicity or an increase in the transmissibility, but you can also have a decrease in the pathogenicity or a decrease in transmissibility. So it's not we cannot answer there is an increase in pathogenicity and transmissibility or a decrease. It depends, and we cannot guess before the recombination. We cannot guess, but it's possible. Yes.
We have a very interesting question here as well. You just mentioned that the virus transmission needs close contacts. However, many publications tell us that SARS-CoV-2 survives in plastic, iron, etc. up to days. How could you explain these contradictory statements?
It's a question of length of time. When I speak about the fragility of the virus, I say that for the naked virus, the virus can stay in the environment, for example, in the sewage, for months. So it's not exactly the same as for SARS. For SARS, there is no possibility that the virus can survive for months in the environment. Maybe it can be days, but not more than few days. So for me, few days reveals that the virus is quite fragile. And if it was months, 6 months, 10 months, 12 months, I would say that this virus is really strong and can persist in the environment for quite a long time.
We will take like 2 other questions. So I see one here from Stéphane Dover, who says, I assume that we also disseminate our own diseases to other species?
Yes, it's exactly possible. We have had, it's not an example in coronavirus, but we have a special example in Africa. The ecotourists that were going in Central Africa to see the gorillas, the mountain gorillas, have been able to transmit the measles virus to the population of gorillas. So it's always possible to for humans to transmit their own viruses, their own diseases, and it's not in a one way. We are mammals like other mammals, and we can transmit our virus to the other animals as the animals can transmit to us their virus.
And now to close and to finish, we will take like 2 other questions that are really practical and that we all wonder about. First, we have a question that says, if we're infected by the coronavirus, what kind of food can we consume to increase immunity and to prevent pneumonia to occur? Or is there any kind of food?
I'm not sure that that the food can prevent the acute infection. I think it's really important that the population can have access to a diversified food, which is rich in vitamin, which is rich in protein, which is rich in fibers. But generally, to be in good health and to have a proper immunity. In case of malnutrition, the immune system can be lowered, and that can be the occasion for the virus to infect the patient. But just for a few times, I think there is no proper food that can prevent the infection with the virus.
Okay.
I think it's a long time process. You need to have a good food, to have a good health, to have a good immunity, to be in good shape.
And a final question that I think, and we all wonder what will happen next. So the question is, how likely will a second wave of COVID-19 happen? COVID-19, sorry.
It's really difficult to say. As I said, the epidemic has just crossed the threshold of 9 million people infected. So it's really unlikely that the epidemic will stop. But in some places where the containment measures are completely respected, it's possible that the virus doesn't spread. There will probably have a second wave of COVID-19. What is important, I think, is that the wave is flattened. I mean, if the epidemic is too strong in a short time, the health system are unable to manage the flow of sick people. So the question is not the question of a second wave. The question is the intensity of the second wave. And what we need to do is to flatten the wave. I mean, if it's a tsunami, it will destroy everything. The health system will be completely out of order. But if the wave is flattened, the health system will be able to cope with all the infected people.
Okay, thank you so much. We have many other questions, but unfortunately it's too complicated to answer to everyone, so we always choose some questions. So thank you, thanks a lot, Thomas Mourez, for your really interesting conference. And could you say a word just to conclude on a positive note, maybe?
On a positive note. Maybe one the positive thought we can have is the possibility of the development of a vaccine. The researchers have proven that the people that have been infected develop neutralizing antibody. And when the people have been infected with the virus, they cannot be infected again. So it means that there is a possibility of vaccine. There is a possibility of development of a vaccine. And what we know is that there are 100 of vaccine candidates that are studied around the world. A lot of them are have been in the process of tests of trial. And I'm quite sure that in 1 or 2 years, maybe less, some vaccines will be available at the surface of the globe, and it will be probably the main factor that will end the COVID-19 epidemic. So I think we will have in 1 or 2 years an effective vaccine against the epidemic.
Okay, we really all hope so. I think the whole world hopes so. Thank you again to Mourez for your presentation. I would like to thank Thierry Marais for being here and the whole scientific cooperation team, Dia and Stephanie. Thank you. And we hope to all see you again for our next webinar that will be coming soon. And thank you again, Thomas, and all have a great day. Bye. Thank you.
It was my pleasure. Bye-bye.
Bye.